In the second section, we'll talk about some other ways to control microbial growth. Usually, if we're talking about trying to kill microorganisms, control the growth, one of the first things that we talk about is heat. Cranking up the temperature is a way to kill microorganisms. Remember that heat is going to denature those proteins. So, if you worked in an industry and you were trying to validate things like autoclaves, these are some of the terms that you might use. So, for example, if you take a look at the last term, decimal reduction time, this is the time required to kill 90% of organisms at a particular temperature. So, for example, if we had a D value 150 degrees Celsius at 30 minutes, that means if we held the temperature at 150 degrees Celsius for 30 minutes, we would actually kill 90% of the organisms that are present. So these terms here, thermal death point, thermal death time, has to do with the temperature and the time points that you would use to kill these microorganisms using heat. Now, what is the best way to heat things up to kill microorganisms? There are different mechanisms. The first one is dry heat, and there are these big dry heat ovens. They're a little slower to penetrate materials. They're really good for metal instruments or glassware, or even things like oils and powders that can't get wet. And this is using a dry heat at a certain temperature and certain amount of time. One of my very first jobs out of college was working at Bausch and Lomb making saline solution. So in our pilot plant, when we'd made gallons and gallons of saline solution, the equipment that we used was metal, and so we used dry heat ovens to actually clean our equipment. The most common thing that we use in labs is moist heat. So we can use moist heat through an autoclave. And what an autoclave does is it's going to heat things under pressure and using steam. Now, this is good for things that can get wet. If they can't get wet, then you might want to use a dry heat oven or maybe some other method to use. So this is most often what we use in a micro lab is an autoclave. So the test tubes that we use in lab have actually been run through an autoclave. They've been cleaned. And if you've gone to the dentist office and they brought out the equipment to use for your dental appointment, it has been run through an autoclave. So I've used these in micro labs. I've used these in pharmaceuticals. And there are different small tabletop, and then there can be much, much larger machines. And so this is probably the thing that we most often use in a micro lab. Early on, we talked about Louis Pasteur and pasteurization. Pasteurization does not sterilize things, so it heats things up for a certain period of time. It will reduce the microbial load, but it won't necessarily sterilize. So when you're buying milk at the store and it says that it's pasteurized, that doesn't mean that it's sterile, that there's no bacteria. That pasteurization has actually reduced the microbial load in that dairy product. And there's different forms of pasteurization depending on the methods and how long they're actually holding it for, as well as the temperature. I'm going to spend a little bit more time talking about the autoclave because this is something that is really common in micro labs. So this picture is an autoclave. In fact, it looks very similar to the one that we have in the prep lab. So our biohazardous waste, for example, is run through the autoclave. Our glassware is run through the autoclave. Even the media can be autoclaved and then cooled and then poured. So this is the setup where you would actually have an access for steam to come in. And this is going to hold heat under a really high pressure. And the door on our autoclave almost looks like the doors that you might find in a ship or a submarine. And it kind of has that big circle that you kind of have to crank. And this is what is maintaining that pressure. It's almost like a pressure cooker, basically, is what we have in the lab. Now, this is what our autoclave looks like. And so here are some other autoclaves that you might see. So the tabletop I used quite a bit in pharmaceuticals. The previous picture was what I used to use in microlab when I was working with the microorganisms that I was characterizing. So when I worked in pharmaceuticals and I worked in formulations, we made some very small batch drug solutions. And so all of our glassware, everything would actually be put through one of those tabletop autoclaves. And it would actually be digital. So it would actually spit out a tape so that we would know exactly what the temperature and pressure was every minute of the run. I used to hear horror stories about doors flying off because they weren't closed properly. And so I will tell you, I was always very paranoid around autoclave. So if I knew that my autoclave was in the other room on one side, I would actually move to the other side of the room just in case something happened with the run. If you look at the picture on the right, this is a big autoclave. So this is something that I saw when I worked at Bausch and Lomb. So we might see this in an industrial setting. We might also see this in a hospital. So they can be varying sizes. If you go to the dentist office, more than likely you're going to see a tabletop autoclave. When we would do our runs, we would actually wrap them up in paper. So the things that we were autoclaving, and then we would seal it with autoclave tape. And then once the run was through, when we would take out the package, that tape would actually have stripes on it. So that was a clue to us that, okay, that had actually been autoclaved. You might see similar type things at a dental office. When I worked at Bausch and Lomb, when we had these huge autoclaves, we had teams, validation teams that would validate our sterilization methods. And so that was their job to make sure that this equipment was doing what it should do. Now, in our lab, we have our autoclave validated, so it is checked on a yearly basis. But if you're unsure if your autoclave is actually performing as it should, you could actually use these little ampules from time to time. And our lab technicians, they will actually put these in an autoclave run occasionally just to make sure that our autoclave is doing what it's supposed to be doing. And this is taking these little ampules that have media, and they also have a little spore strip. And so because this autoclave is under pressure, when this ampule is put into a run, the pressure will actually squeeze the tube and break the internal glass tube. And it allows the media to actually go into that spore strip. So after your run, you would actually take this out and then you would put it in an incubator. If you saw no growth, then that means that your autoclave was doing its job in sterilizing and it killed those spores. If you saw growth and it was cloudy, that means that your autoclave really wasn't working as it should. So it wasn't a high enough temperature and pressure in order to kill those spores. There are some other methods than heat that we can use to slow down bacterial growth. Refrigeration. Now we know this with our kitchen. So if you have leftovers, you want to put them back into the refrigerator because that lower temperature is going to help to slow microbial growth. So if we want to preserve some specimens, sometimes if I'm trying to save plates, I'll put them in the refrigerator. Now, these are living organisms that sometimes when we put them in the refrigerator, they don't always look and behave the way that we want them to, but this is something that we certainly do in our own homes. Put it in the refrigerator, and that can help extend the life of your leftovers. Freezing is another thing that we can do. So we can freeze our leftovers. You go to the store and you get some chicken, and then you can actually put it in the freezer, and now it can be good for months. We can actually freeze things down to a very low temperature so that we can actually preserve our microbes. So this is something that they can do in micro labs just to make sure that that there's going to be a supply of microbes. Anytime that you take the water out of something drying, that means that it's going to inhibit microbial growth. Remember, though, that our endospore forming bacterial friends they can actually survive that drying. So may not necessarily be the best option for everything. And then freeze drying. This is drying something after it's frozen, also called lyophilization. If you've ever had space ice cream where you have those little pellets that are cold, the little ice cream pellets, that's basically lyophilized ice cream. One thing that we will be testing in lab is radiation. So UV light can actually kill microbes, so it can damage the proteins, can also damage the nucleic acids as well. UV light is one of those things that certain wavelengths can actually get rid of viruses and bacteria. So we're going to be testing UV light in lab. We're actually going to be using a microorganism that has a pigment, and that pigment actually helps to absorb some UV light. So it's going to allow us to actually see different time points in how UV light exposure affects that microorganism. UV light is good at getting rid of both viruses and bacteria because it damages nucleic acids. However, it's not so great at getting through things. So it's useful for decontaminating the air. So I mentioned our biosafety cabinets. Obviously, when we were working in those cabinets, we used visible light, but at the end of the day, once we were done and we cleaned it, we actually flipped on another light that was UV light. So it actually killed anything on the surface. UV light is not so good at getting through things. So if you were to take a plate of growth and put the cover on it, or even a piece of paper and expose it to UV light, it's not going to be able to get through. So it's not going to be able to do anything to that organism. It's the same principle of why we wear like sunglasses. So those sunglasses help to protect our eyes from UV light because it's not so good at getting through things. Gamma radiation is another thing that they will use, not only in different industry like pharmaceuticals, med tech devices, but they will also use this for food as well. I've also seen it in the makeup industry as well. So this uses X-rays and gamma rays that actually creates ions and it dislodges electrons. This uses cobalt-60. Many industries are actually trying to find some different ways of sterilization because there's, from what I understand, a world shortage of cobalt-60. This is very useful for getting rid of bacteria and viruses in foods, different products, medical devices. So my husband works for a medical device company, and they actually design equipment that you would use in surgeries. He actually designs the packaging. And so once that device is packaged up, they will actually send it through gamma radiation, and that will kill anything in there so that it's actually sterile when it gets to the surgical suite. I've also seen it used in foods. Many grocery stores might label this and actually even put it in a little different display cabinet. So I was actually going to a grocery store one time to buy some burgers to cook on the grill. And they had the regular burgers. And then in another case, they actually had preformed burgers that had been hit with gamma radiation. Now, this does nothing to the food. It does nothing to the people that are eating it. It just has to do with killing those microorganisms. So if you haven't gathered, I am a germaphobe. So I was happy to buy those burgers that I knew had been run through gamma radiation. So you might see this in different industries. Microwave radiation, this can also sterilize things. Only useful if it contains water. And strong visible light can also affect bacterial enzymes. Certain dyes can actually be more effective with strong light on microbes. So here is a picture of the effect of UV light on bacteria, which we will be testing in lab. And so here we have a lawn of growth, and it's been exposed to UV light. So you can see the growth, you can see those colonies. before the UV light was actually centered on this Petri dish, before this organism was exposed. And then after, you can see that it actually killed the bacteria. We are going to be testing different time points and how long it's going to take for that UV light to actually kill all of the organisms and what happens with the exposure time. Again, we're going to be testing this on a special microorganism, Serratia marcescens, that produces a red pigment. And that red pigment helps to absorb some of the UV light so that we can actually see those the different effects with the time points. Some other methods, ethylene oxide gas, or ETO. This is an alkylating agent that disrupts cell metabolism. They can actually use this for breathable packaging, so if it can actually get in there. Again, it's not going to do anything to the equipment. It's just going to sterilize it, get rid of the microbes. E-beam, or electronic beam. This is something that they've been using more of because of that shortage of cobalt-60. This uses beta particles to inactivate microbes. It is faster than gamma radiation, but it doesn't penetrate as much. And then chlorine dioxide gas. This is something that's been around for a while, but again, they've started to use this more. It's a strong oxidizer, so they can actually use this for food, public water supplies, medical devices as well. Now, you might be thinking, wait a minute, chlorine, it actually does not chlorinate. So it's not what you would typically think of. And so this is another method that they've been using more and more. This last, one of the last things that we'll talk about is filtration. So if we need to sterilize a liquid, but we can't autoclave it, we can't heat it up because it will damage it, then we may want to use filtration. And filtration is using these filters with tiny, tiny pore sizes. It basically filters out any infectious agents. Now, I told you about my bacteria. They were halophiles. So they like salt. So I used a media that they liked, that was food for them, that would allow them to sustain their life. But every time I added salt to it and then autoclaved it, the salt would actually precipitate out into a solid. So I was not having luck preparing my media in an autoclave. So I ended up having to go to a filtration system. So I filtered my media to sterilize it because I did not want to contaminate my bacteria. And those pore sizes were small enough to filter out any infectious agents and contaminants. So I've also used this in pharmaceuticals. So I mentioned formulating drug solutions. So we would autoclave our glassware, but when we were formulating our drug solutions, we could not heat them up because we would end up destroying the drugs. So we use these very tiny filters. Now, when you buy these filters, they can be very large for large scale production or very small. I mostly use the small ones. So you can see on the right is a very small filter that's actually attached to a syringe. So in my biosafety cabinet, I filtered my drug formulation through this filter and then collected it in my sterile glassware and then sealed it up. We could not heat our drug solutions because they would have been destroyed. What you're seeing on the top left, those are the containers that I actually used to sterilize my media. So this came all wrapped up in sterile, and it actually came with a sterile collecting vial. And I would do this in the hood. So I would pour my media in. If you notice that nozzle, I could actually attach a hose up to a vacuum system. I would turn on the vacuum system. And when I added my media to the top, it would be sucked through that filter and filtered out any contaminations so that I had sterile media that then I could cap and I could store and I could use for a period of time. So again, they can be very small. They can also be very large for large scale filtering. The last thing that we've already talked about in some previous chapters, this is osmotic pressure. So this has to do with the tonicity, so the amounts of solutes and water we have on either side of the membrane. And I mentioned how these high concentrations of sugar and salt will basically draw the water out of the organisms. If the water leaves, bye bye bacteria. It's not going to be able to live. And so this is something that they've done to preserve food for years and years. So in like salted meats, jellies, jams, because of that osmotic pressure, it draws the water out and then the bacteria can't live.